Welcome to Cell and Molecular Biology. Today, we are going to explore one of the most fascinating and essential processes in all of life, prokaryotic DNA replication. If you have ever wondered how a single microscopic bacterium can multiply into millions in just a few hours, the secret lies entirely in how fast and accurately it can copy its genetic code. We will build your understanding from the ground up, looking at the biology, the real-world applications, and even the mathematics behind this incredible microscopic machinery. Let us dive in. To begin, let us imagine you have a massive, ancient recipe book that contains all the instructions needed to run a highly successful bakery. Now, suppose you want to open a second bakery across town. You cannot just tear the book in half. You need to copy every single word perfectly so both bakeries have the full, exact set of instructions. In the biological world, a bacterial cell faces this exact same problem right before it divides into two new cells. It must copy its entire recipe book, which is its DNA, so that both the mother and daughter cells have a complete set of genetic instructions to survive. This copying process must be incredibly fast, but also incredibly accurate, because a single mistake could result in a defective, dying cell. Now that we understand the basic goal, let us look at the formal biological definition. Prokaryotic DNA replication is the process by which a single-celled organism, like a bacterium, duplicates its entire circular double-stranded DNA genome. We describe this process as semi-conservative. This means that when the copying is done, each new DNA molecule consists of one old, original strand and one newly synthesized strand. It is also bidirectional. Instead of starting at one end and going to the other, the copying starts at a specific single spot called the origin of replication. From there, it moves outward in both directions simultaneously, which makes the whole process much faster. Building on that bidirectional movement, we must also understand that the process is semi-discontinuous. Because of the chemical structure of DNA, the two strands cannot be copied in the exact same way. One strand, known as the leading strand, is copied continuously in a smooth, uninterrupted motion. However, the other strand, known as the lagging strand, has to be copied in short, fragmented pieces. We call these pieces Okazaki fragments, named after the scientist who discovered them. This semi-discontinuous nature is a direct result of the physical constraints of the DNA double helix, requiring the cell to use a clever workaround to copy both sides at the same time. To manage this complex, semi-discontinuous copying process, the cell relies on a highly coordinated team of enzymes. You can think of these enzymes as workers in a microscopic factory, each with a very specific job. First, DNA helicase acts as the unzipper, separating the two original DNA strands so they can be read. Next, primase comes in to lay down a short starting primer, which acts like a flag, telling the main builder where to start. Finally, DNA polymerase III, the star of the show, reads the original strand and builds the new DNA by adding the correct nucleotides. Together, they ensure the new strands are built efficiently and accurately. Let us visualize everything we just discussed by looking at this diagram of a replication bubble. The large blue circle represents the original prokaryotic circular DNA. At the top, you can see the origin of replication, labeled ORI-C, where the process begins. Notice the two replication forks moving away from each other in opposite directions, shown by the red arrows. This perfectly illustrates the bidirectional nature of the process. At each fork, look at the newly synthesized red DNA. One strand, the leading strand, is built continuously. The other, the lagging strand, is built in those short Okazaki fragments we mentioned. This single image captures the semi-conservative, bidirectional, and semi-discontinuous mechanics of the bacterial copying factory. Having seen how the machinery works, you might wonder why this matters in the real world. Understanding prokaryotic DNA replication is absolutely critical because it is the fundamental basis of bacterial growth and survival. Without this process, bacteria could never divide. In modern medicine, this knowledge is a cornerstone for developing antibiotics. Many life-saving drugs, such as fluoroquinolones, work specifically by jamming this exact bacterial DNA replication machinery. By stopping the enzymes we just learned about, these drugs prevent harmful bacteria from multiplying in your body. Because bacterial enzymes are structurally different from ours, scientists can design these drugs to target the bacteria without harming our own human cells. Now that we understand the biological importance, let us look at the mathematics. Although biology often feels descriptive, this process is highly mechanical and moves at a measurable speed. We can calculate the exact total time it takes for a bacterium to copy its entire genome using this formula. T equals N divided by the quantity 2 times R. Here, T is the total time in seconds. N represents the total number of base pairs in the genome, which is the total physical work to be done. R is the replication rate, or the speed of a single DNA polymerase enzyme. We multiply R by 2 in the denominator because the process is bidirectional. 2 forks are working at the same time, cutting the total time in half. If the genome size N increases, the time T naturally increases. If the enzymes work faster, increasing R, the total time T will decrease. Let us apply this formula to a real-world scenario involving the famous bacterium E. coli. We are given that the E. coli genome has 4,600,000 base pairs. We also know its DNA polymerase operates at a steady rate of 1,000 base pairs per second per fork. We want to find the total replication time. First, you write down our formula. Next, we substitute our known values. N becomes 4,600,000 and R becomes 1,000. The denominator becomes 2,000, accounting for the 2 replication forks moving in opposite directions. Dividing the total base pairs by the combined speed gives us 2,300 seconds. To make this more intuitive, we divide by 60, giving a final answer of approximately 38.3 minutes. Physically, this means an E. coli cell can completely duplicate its genetic manual in under 40 minutes, explaining why bacterial infections spread so quickly. This incredible speed brings us to another mathematical reality: the chance of making a mistake. DNA polymerase III is an incredibly accurate molecular machine, but like any physical process, it is not absolutely perfect. We can calculate the expected number of genetic mutations introduced during one cell division using a simple multiplication formula: E equals N times epsilon. Here, E represents the total expected number of errors per replication cycle. N is again the genome size in base pairs, representing the total number of nucleotides that must be copied. The Greek letter epsilon represents the error rate of the DNA polymerase enzyme, which is usually a tiny fraction. By multiplying the total amount of work by the rate at which mistakes happen, we find the total expected mistakes for the entire copying job. If the genome is larger or the enzyme is less accurate, the number of errors will naturally increase. Let us see how many mistakes actually happen in our E. coli cell. We know the genome size, N, is 4,600,000 base pairs. The known error rate, epsilon, is approximately one mistake for every 10 million base pairs synthesized. We want to find out how many errors occur in one complete round of cell division. We take our formula and substitute the values. We multiply the 4,600,000 new base pairs that must be added by the fraction 1 over 10 million. The calculation yields a final value of 0.46 errors per replication cycle. Physically, this means that on average, an E. coli cell makes less than one single mistake every time it copies its entire genome. This delicate balance between perfect copying and rare mistakes allows bacteria to maintain stable identities while still driving evolution. We have covered a lot of ground today, so let us summarize the key takeaways. First, we learned that prokaryotic DNA replication is a semi-conservative, bidirectional, and semi-discontinuous process. Second, we saw how a microscopic factory of enzymes, including helicase, primase, and polymerase III, works together to copy the genome. Third, we discussed how targeting this machinery is the foundation for many life-saving antibiotics. Fourth, we used mathematics to prove that replication time is directly tied to genome size and enzyme speed, allowing bacteria to multiply rapidly. Finally, we calculated that the incredible accuracy of these enzymes ensures genetic stability, while still allowing the rare mutations that drive evolution. You now have a solid, intuitive, and mathematical grasp of how bacteria duplicate their DNA. Great work.